This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 415, recorded on November 9th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Southeastern Michigan, Kathy Spindler.
Hi, everybody.
Everyone's a bit muted today.
Yeah. I didn't think the sun had come up today, but finally the clouds are white instead of gray, and there is some blue sky, and the sun is up.
Yeah, we have no sun. We have clouds and rain, 14 Celsius here. Kind of a gray day.
Yeah.
We have a big crowd today. Let's see. No longer from North Central Florida, from Austin, Texas, Rich Condit.
Hey, everybody. How you doing?
That's right. You're in Austin now, right?
I am in Austin, Texas.
And
I no longer own a house in Florida.
Congratulations.
As a matter of fact, I'm homeless because the house here isn't done. So we're camped out with my daughter and her family for almost 2 months.
Well,
which is going to be a nice test of this whole thing, but we get along well. So it's actually been rainy and cloudy for a week here in Austin, and today it's 63 degrees right now and cloudy. It rained a bunch last night. It's rain possible today.
Well, congratulations on the move, and I'm sure.
Thank you.
I'm sure in the future I'll say North Central Florida because I did that for so many years. So
that's okay. I'm going to maintain my professional and emotional affiliation with the University of Florida. I'm still an emeritus professor there. So is Austin in North Central Texas, or is it? I think it's kind of Central Texas.
Central Texas. Okay.
Yeah.
I'm not looking at a map.
Well, I will look at a map and make a judgment.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. So it's pretty much the same weather you have in New York. It's 8 Celsius. We got a little light rain falling, kind of a gray, blah sort of day.
It is indeed. But we'll cheer everyone up with stories about viruses.
Yes.
Now we have 3 guests on TWiV today. I don't know if we've had 3 guests. We've had 2 guests, but we have 3 today. And the first, is it Central Massachusetts? Jeremy Luban, welcome back.
Thank you, Vincent. I guess we could call it Northeastern Worcester.
Northeastern Worcester. From Worcester, Massachusetts.
Yes.
Jeremy is back, and you were here, I don't know, a couple of weeks ago, it seems, right?
Yep.
Talking about.
I think Worcester is usually considered the beginning of Eastern Massachusetts.
Okay. From Eastern Massachusetts. Well, welcome back, and today we're going to do another story from your laboratory, a very different story. So to do that, we actually have 2 of the participants in the work. One of them is back for a second TWiV. He's also in Worcester, along with Jeremy. Ted Diehl, welcome back, Ted.
Hello again, everybody.
Did you think you'd be back on TWiV so quickly?
Not this soon, no.
It's pretty good.
Everything happened quite quickly.
Well, that's good. And you're a postdoc, is that right?
I am a postdoc, yes. 2
TWiVs. Now, the one, when you were with Welkin, were you a postdoc or a PhD student there? I think you're a PhD student, right?
No, I was a postdoc. It was my first postdoc with Welkin. Okay. This is my second with Jeremy.
2 postdocs, 2 TWiVs. I think that's pretty good. Congratulations.
Yeah. Thank you.
And our other guest is at the Harvard University Virology program, which is in Boston, of course, and at the Broad Institute of Harvard and MIT. Aaron Lin, welcome to TWiV, Aaron.
Hi, thank you so much for having me. I'm excited to be here.
Well, thank you for coming on. Of all of the people on TWiV, you're the new one, and we'll find out a little bit about you in a moment. But I want to tell everyone first that the Department of Microbiology at the Icahn School of Medicine at Mount Sinai here in New York City, they want you to know about their graduate and postdoctoral programs. Their department is composed of over 20 virology labs, all centralized in one building in the heart of New York City. The department's a perfect fit for anyone with an interest in pursuing virus research. The department is presently looking to recruit prospective graduate students to apply to their program by the December 1 deadline. You have a little bit of time, but not a huge amount. Interested postdocs are also encouraged to contact faculty of interest. For more information about the department, visit bit.ly/micromssm. That's all one word, MICROMSSM. Just one little update before we move into today's stories. We've been getting lots of entries in the Limerick Haiku Contest. It's great. You're doing a great job. And next week, we'll pick a winner. So you have until November 14th to get your entries in. I've gotten a few this week already, and this has been now a couple of weeks that we announced it, right? So people listen weeks and weeks later. So November 14th will be the last time you can send it in, and then we're recording early again next week. So we'll announce our winner next week. But you're doing a great job. Some of them are so awesome. It's just going to be hard to pick a winner. That one. So that's cool. Now, the reason for us having three guests today is because they have together published a paper last week in Cell, and it's called Ebola virus glycoprotein with increased infectivity dominated the 2013-2016 epidemic. And this has hit the news. And the first two, the co-authors, are Ted Diehl and Aaron Lynn. And way at the end, after many other authors, is Jeremy Luban. And some of the authors in between are Grubaugh, Carvalho, Kim, Kiawe, Macaulay, Donard, Kukural, McDonald, Schaffner, Garber, Rambeau, Anderson, Savetti, and Luban. A lot of authors there. So I heard about this story a couple of weeks ago, and I said, well, Jeremy, when it's published, let's get you on. So here we are. And we'll talk about this. There's also a companion paper by another group, which we will mention, of course.
Yes.
But not go into it in any detail, because I'm sure we'll go into a lot of detail on this one.
In fact, when Vincent and I heard about this from Jeremy...
Oh, that's right. You were at the same meeting.
We were then... I remember, Jeremy, I was sitting next to you as you were sitting through a talk by the other group, and you were frantically texting with your collaborators on the paper about the overlapping data, shall we say.
Yeah, in fact, that may be another group.
Okay.
So
yeah, it might be a third group.
Yeah.
All right, so before we talk about the science, Jeremy, I want Aaron, tell us a little bit about your history. Where are you from?
So I am from the Bay Area in California, from a little town called Sunnyvale. I grew up there, went to high school, but then went to the East Coast for college, actually. So I went to Princeton and kind of had an interest in viruses even a little bit before that, but then took Lynn Enquist's virology class at Princeton and really loved it. It was really incredible. So that sort of cemented my interest a lot. I actually worked in the research lab of Ileana Cristea, who's been on TWiV a couple times now, including a video interview. I did a lot of proteomics, looking at protein-protein interactions of viruses and their cognate host proteins. And eventually went on to graduate school here at Harvard in the virology program. After a couple of rotations, I have settled into the lab of Party Savetti, and I couldn't enjoy it more. It's been really great. We've got a lot of really lovely collaborations, a lot of great science happening, and a lot of different, very exciting topics. So, really, just sort of glad to be part of the whole grand scheme.
And you've heard of TWiV before, right?
Yep, I've been listening since pretty much Lynn Enquist's virology class, which was maybe 4 or 5 years ago at this point. So I'm really excited to be on the show.
Great.
This is a lot of fun.
Cool.
All right. So, Jeremy, just sort of like you did last time, why don't you tell us, the backstory here of this work, and we can let Ted and Aaron fill in the details as we go along.
Okay. Excuse me. I guess we're all a little subdued today.
We are.
It's kind of a strange day.
Yes.
Post-election. Let's see. I guess Ted was already on, so he went through. I think on the previous episode he went through his background.
Right.
Let's see. So when I was last on TWiV, we talked about HIV-NEF, this mysterious protein that does everything.
NEF is enough.
Right.
NEF is enough, right?
Right.
NEF is enough. Yes.
And how NEF seems to inhibit an antiviral factor that's present in human cells called serine, or serine-5 in particular seems to be the family member with the most activity. But there are others in the family with activity. But we've focused on serine-5, and serine-5 is a membrane protein it's incorporated into the envelope of the new virion particle as it's formed. And if it's present there, it inhibits the infectivity of the particle when it goes on to infect a new target cell. And one of the things that was clear from the experiments that Massimo Pizzato did, and that also information that had accumulated over the 20 years prior to that about the effects of NEF. One of the things that was clear was that whatever NEF was doing, it had something to do with the glycoprotein. At least that as far of the viral components, the glycoprotein seemed to be a critical determinant of how potent this antiviral activity was. So there are HIV-1 glycoproteins that are more sensitive and glycoproteins that are less sensitive to the effects of serine. So once it was found that serine was this missing factor and that serine was incorporated into the virion, an obvious question was whether serine was an effective inhibitor of other viral glycoproteins that are encoded by enveloped viruses that are driving entry into new target cells. And so Massimo and Heinrich as well, and then Ted, when he started working here in Worcester, started looking at different glycoproteins from creatures that were not HIV-1. So Ted has embarked on a very large survey. He's looked at the broadest range of retroviruses, murine oncoviruses, and Mason Pfizer monkey virus, and all different kinds of retroviral glycoproteins. And the question being, how many of these viruses encode glycoproteins that in effect are sensitive to the effects of serine? I should maybe point out that we're still not certain how serine works. We think it acts late in the fusion process, maybe after hemifusion, when an initial pore is established that has to then expand to release the full contents into the cytoplasm. So you can imagine that the effects of serine would vary quite a bit depending on the exact geometry of the glycoprotein or the exact mechanism that it uses to fuse. Does it fuse at the surface? Does it fuse deep in the cell? And in fact, one of the hypotheses that Ted had was that serine might only be effective if it acts on glycoproteins that fuse at the surface and are not dependent on the acidification that occurs, say, with other viruses that have to travel deep in endosome compartments and where the pH drops, which then enables conformational changes that allow fusion. So initial experiments with Ebola, which is one of these deep traveling glycoproteins, that is Ebola, to fuse, has to bind to the surface, has to be endocytosed, and the whole virion has to be transported deep into the cell within an endosomal compartment. And the pH has to drop quite a bit, say down to pH 5, before fusion will occur. With initial experiments with Ebola, with one of the standard glycoproteins that are available, no effective serinc was seen.
So that's something that you did, Ted, that experiment, right?
Well, I'll just interrupt and I'll let Ted take over, but I'll say from the initial experiments, even with Massimo, some of these viruses were looked at, and one had the impression from these initial experiments that it was really a retrovirus-specific kind of phenomenon. But then Ted looked in more detail, and I'll shut up at this point and let Ted take over.
No, I think you're doing fine. But
I'm curious.
I was just going to jump in and say that it was Massimo who first made that observation. But yes, I've done the experiments, and it holds up.
So initially, what Ebola sequence did you use? Because there are lots of possibilities, right?
Yeah, it was the 1976 Myanga strain.
And you do this by making pseudotyped lentiviruses, right?
Correct.
So maybe you could just briefly explain what that is.
So we transfect together the GAG-PAL structural enzymatic plasmid, makes everything that's necessary to make viral particles that are able to get into a new cell, reverse transcribe one time, integrate, and then express something. So the ones that we're using have a heterologous promoter, like CMV in this case, and GFP. So you can see which cells are infected. And then into that, we throw in different glycoproteins. So typically we'll use VSV because it gets into pretty much everything. But for these experiments, we used a whole host of different viruses.
Okay, so 1976 Ebola glycoprotein.
And a host is the correct collective noun for viruses.
1976 Ebola glycoprotein, no effect of serinc, right?
None whatsoever.
All right. So then what happened next? Something must have happened.
Well, I think I should turn this back over to Jeremy because I'm not actually sure where and how getting our hands on the other constructs came about.
Well, so we decided together that Ted and I that we wanted to collect. Well, actually, maybe this occurred before you got here. I'm trying to remember.
This did occur before I got there.
Yeah. We, meaning the Royal we, wanted to do a survey of different glycoproteins. I mean, it seemed kind of, well, serinc was very effective at blocking HIV, and it would be kind of cool if serinc was effective against a wide range of viruses. And there's a lot of envelope viruses out there if you think about it. So we started collecting glycoproteins, and Ebola, because of its prominence and as being such a nasty virus, and the recent epidemic in West Africa, we decided that we wanted to go beyond the 1976 strain that was tested and look at all the different types of Ebola, which the nomenclature is very complex, and I'm sure I'll make lots of errors, but they're analogous to different species of Ebola.
Jens Kuhn will correct you if he hears you make.
Yes, he's
right.
He's.
I'm sure you'll be getting more than one follow-up email from him.
Yeah, we were on the phone yesterday. In fact, he's taught us a lot about nomenclature. So, yeah, there's the Ebola restin that doesn't seem to harm people as best we can tell, but can cause very severe disease in certain animal species. There's a virus that was isolated from a bat in a cave in Spain that we don't really know too much about, as far as I know. And then there are a couple of types that are quite deadly in people. from Africa. So we collected glycoprotein representatives of each of these when we could. So this was a lot of cloning by phone, or now by email, as we call it. Some people readily send you things. Some people don't really know what they have, and they send you what they have, and then you have to sequence to figure out what it is. Some people require that you jump through all kinds of hoops. It's a process that's probably familiar to a lot of listeners who are working at the bench today. And then one day there was a talk here on our campus by a scientist who I gather has an alternative career as a hair model. I learned from Twitter. His name is Christian Anderson, and if you look him up, you'll see what I mean. But Christian gave this amazing talk about the West African outbreak. And I met with him, and we talked after his talk about what my lab does, and through Christian, we found out that they had isogenic glycoprotein clones, plasmids, for a kind of ancestral form that was thought to have established the outbreak in West Africa, with point mutations on top of that. And these were point mutants that phylogenetic analysis suggested might be interesting. And so these constructs were engineered by a very talented student in Participetti's lab. Christian at the time was kind of a super postdoc there. He now has his own group in San Diego. So that student was Aaron Lynn, who's here. And so we collected a couple of these things and we tested them. And I think eventually Ted arrived in the lab. I think it's kind of interesting to think about the fact that Ted was here working for maybe 2 years on a totally unrelated project about something called HERV-H. I don't know, Ted, you want to pipe in?
I don't know how relevant it is for this story, but yeah, it was about a year and a half that I was working on HERV-H, and had the IRV paper come out of Welkin's lab during that time. But
I think it's interesting in that that project, which is still something we're very interested in, was extremely difficult for technical reasons, and was really unable to move very far. When all of a sudden this potential project with Surinc arose, and Ted decided to take a look. Maybe it was really inspired because we had a rotating student, and we were looking for a project that wouldn't be too difficult technically. Is that right?
Yes, yeah, that started with PPK, who's on the paper.
PPK.
Yeah, nobody can pronounce his actual name, so he goes by his initials.
I think that's actually an enzyme of some kind, right?
Yeah, it could be.
I think so.
I think it's also James Bond's gun.
Yeah, Walter PPK.
Yeah,
right?
Walter PPK. Yeah.
So I want to I just want to pipe in and say, you said HERV-K. It's a human endogenous retrovirus that he was working on, that Ted was working on, right?
Right.
Correct, but it was HERV-H.
HERV-H.
Did I say K?
No, I'm sure you didn't. Yeah. I just wanted to tell people who are saying, what the heck is HERV?
Yeah, HERV, human endogenous retrovirus type H, which we believe is involved in pluripotency.
Ah, yes. We did your paper a long time ago on that.
Yeah. So anyway, so indeed, attempts to determine whether Surinc 5 had effects on pseudotypes with all of these different Ebola glycoproteins were, in fact, a bust. That is, serine 5 had no effect on the infectivity of any of these things. But then Ted kind of pays close attention to the data he generates. I've noticed it's kind of a nice skill to have, the ability to pay attention.
Or a curse, depending.
Yeah. And so he noticed that among the variants that Aaron had sent to us, all was not equal in terms of infectivity, and that's how this story arose from our standpoint. Again, I don't know. Do you want to take over, Ted, or should I keep going?
I'm fine for you. Keep going.
So at this point, let me understand this. So, Aaron, you made these constructs, the glycoproteins with the various sequences. Is that correct?
Yeah, that's correct.
Were you doing? You were doing something with them in your lab, I presume, right?
Yeah. So we'd actually been eyeing these for quite some time. I think you guys covered this on the show a while back, but our group had published kind of in early summer of 2014 some like really early next-gen sequencing data on the outbreak as it was happening almost in near real time in Sierra Leone, when the number of cases was really starting to accumulate. Pardis has had really good collaborations with Kenema Government Hospital in the region, as well as other hospitals nearby, and so we started to collect these excesses of cases that were coming in and tried to understand a little bit more about how the virus was spreading between people to people. Obviously, a lot of it, even early on, was thought to be due to infrastructural issues. But of course, we always want to, make sure we're like looking through every nook and cranny to make sure there's not some other genetic changes that are occurring. And so, we had done some of the sequencing of these Ebola virus isolates. And one of the really striking things that had popped out of even the early phylogenies was that, there's a few mutations that had become really prevalent in Sierra Leone. Now, at that point, it was a little bit harder to say too much about them because there wasn't a lot of other sequence data coming out from the other two countries that were mainly affected, Guinea and Liberia. And we only had sort of our bit of the picture in Sierra Leone. But even from just that kind of data, like one of the ones that we thought would be interesting to follow up on was this glycoprotein variant, just because the glycoprotein's relatively well characterized compared to a lot of the other Ebola virus proteins. And on top of that, there's a lot of good systems for working on it in BSL-2 settings. So we'd kind of started to put together some preliminary attempts at functional characterization of this variant, as well as a few others that looked like they might have risen to higher frequency than you would expect by chance. Mm-hmm. And so I think I kind of cloned these out. And at that point, Christian Anderson, who Jeremy mentioned, had come back to me and was like, hey, we found this really great, I found this really great guy at UMass Med. He's doing some awesome work. Jeremy Luban was like, oh, I've heard of him before. It's like a, big virologist in the field. And so he connected us, like Jeremy mentioned, and we're always happy to kind of share any sorts of reagents we made and that sort of thing. Sorry. Sorry, Pardis is popping in for a little bit. And yeah, so that was kind of the story from our side, and we were really happy to work with Jeremy and kind of support them, especially sort of with the epidemiology. As more and more sequences came out, it became really clear that this glycoprotein variant just kind of kept propagating pretty continuously through many transmission chains.
So, Ted, can you tell us what Jeremy mentioned, that you pay attention to your data? Tell us what that means.
So typically for these assays, when you're looking at Syrinc, I don't want to say hyper-analyze the data, but you process it pretty thoroughly. And in the end, you're not really looking at raw infectivity. You're looking at a ratio of infectivity with Syrinc and without Syrinc. So you get some ratio. If it's near 1, there's not really an effective Syrinc. If the number is super huge, there's a big restriction. But if you look at the raw data, you can get 10 cells for the pseudotype, or you can get 50% or more of your cells with a different pseudotype. So I sort of handed this off to PPK during his rotation, and he was just sort of, I told him how to do the analysis for Syrinc, and he did all that, and that was fine. And it wasn't until the very end of his rotation that I started, asking questions, as one might do when they're thinking about putting together talks and stuff like that. And I asked him about the raw infectivity. We went over the raw data, and it became clear that at least for the majority of analyses that he did, there was a pretty glaring twofold phenotype for this V82 substitution in the glycoprotein. So I think he did it 3 or 4 times, and it was repeatable all except for one, and that one was his first time, and it was his first transfection that he'd ever done. And there was a whole bunch of caveats. So I thought that was pretty interesting to have a rotation student who'd never really done tissue culture be able to get a twofold phenotype with a single point mutation in the glycoprotein. That really suggested to me that there was something to it.
We talk a lot about serendipity on this show, and if I'm understanding this correctly, what I'm hearing is that Aaron, you were looking at a bunch of different sequences and noting that during the Ebola epidemic, some seemed to become fixed in time, but the implications of that were not clear immediately. And Ted, you were using those same sequences for an entirely different purpose, okay? And noticed a difference in infectivity. So you have 2 parallel investigations, all right, that in the end come together. When you read this paper, it looks like, oh, here's a sequence in the glycoprotein that comes up with time. I wonder if that doesn't influence the infectivity of the virus, okay? But no, that's not how this happened. This happened as more or less a surprise.
Yeah.
Yes, you started off with a very clear plan to do nothing like this.
Right.
Exactly.
Yeah.
Exactly. No, and to me, this is what science is all about. This is the beauty of it. There was a moment when the 2 stories collided, the Syrinc story and the phylogenetic analysis, and I remember quite clearly that story. When that happened, we had a video conference, a teleconference. Ted and myself here in Worcester, and Aaron and Pardis were in Boston, and so we could see them quite clearly. Pardis has this camera, so you can see her whole office, and we said. So Ted went through the papers, and he had a hunch that there was something kind of interesting going on by this time. And so we said, sort of naively, is there something interesting about this alanine 82 to valine change in the glycoprotein? Because it seems to be more infectious. And Pardis tends not to sit still very much, but I remember she was a little more kinetic than usual in response to this. And so they were really excited. And it was one of those magical moments in science.
Yeah, unfortunately, the papers are written always differently. You never get the backstory, but that's what we can do here on TWiV, right?
Yeah. That's why the backstory is so much fun. So look. I'm jumping into the paper here, looking at figure 1. The thrust of that is that this particular glycoprotein mutation shows up in a fashion that appears to correlate with the epidemic really taking off. Aaron, that was your observation, right? Or does that come after the fact as well? How did that arise?
That actually happened really early on. So like I'd mentioned, it was interesting because when I started rotating in Pardis's lab, that was still my first year. So, we go through a few rotations to test out labs. And it had just so happened that I'd started rotating essentially as the outbreak was spreading from Guinea into Sierra Leone and Liberia, kind of around like spring 2014, essentially. And so as they're generating the data, I was actually working on a different project. I was working with Christian Anderson on Lassa virus, not Ebola. But, I'd see the data come in through lab meetings, and just to kind of watch, just to be able to look at all the sequence data and see how other people were analyzing the data. This was just one of those. Most of the mutations that arise are either unique to maybe a handful of sequences, or they tend to be synonymous or non-coding, and so it's a little bit harder to get a handle on what's happening with those. But in this case, because it was a non-synonymous mutation, meaning it changed the amino acid, and it was in the glycoprotein, which is, got some pretty well-defined phenotypes like entry and infectivity, antibody escape. This was something we wanted to keep an eye on. So it definitely wasn't just my observation alone. But it was something
but you were
that
watching
I started
it.
to think about.
This wasn't even retrospective. You were watching this happen in real time, right?
Yeah, because if you look at the sequences in terms of their geography as well as the timing, the ancestral allele, meaning the having the alanine in the glycoprotein at that position 82, is pretty much present. If you look now that we have a lot more of the sequence data, it's pretty much present almost exclusively in Guinea. And then the strains, the variants that were spread to Liberia and Sierra Leone were almost exclusively had the derived allele, meaning the valine at that position. So once we started sequencing cases from Sierra Leone, they're all valine 82 at that position. So that's pretty striking.
What's amazing about this too, this epidemic in particular, is that the technology has evolved to the point where this sort of sequence gathering is possible, and you can actually talk about, I guess, allele frequency, right, in the population, how frequent this thing shows up, which is an important quantity for informing you as to how important this might be.
Right.
And in fact, even more than that, it's not just the allele, but it's if you look in that first figure, you're actually looking at 1,400 plus near full genomes. This is 19,000 nucleotides that are used to establish the phylogeny. So it's not just amplifying a little region and seeing is this mutation there or not, but it's an incredible collection of data in real time from an outbreak. And
so relative.
Go ahead.
Relative to that, when did this observation of increased infectivity occur? Ted?
This was right around the time that we were taping my TWiV episode.
So
it was right around March. Yeah.
We should.
Okay, so the Ebola epidemic was basically gone by then.
Yeah, it's history according to TWiV.
Right.
So by that time, the Ebola epidemic was over, right?
Yes.
For all intents and purposes, yes.
Yeah.
All right.
Yeah, I think there were still a few lingering transmission chains at that point. But yes, the bulk of the cases were decreasing.
So this change we're talking about in the glycoprotein A82 to V, alanine to valine, according to your paper, it says it was first sampled in Guinea in March 2014. So how many cases were there by that time, roughly? I don't know. Aaron, do you know, Aaron?
I don't have an exact number offhand.
It was not thousands yet. It was maybe hundreds.
It was, it was
a couple hundred.
Definitely not. It was a few hundred, I think.
Okay.
That sounds about right.
Now, Ted, when you observed this effect, what was the next step?
Well, when I observed it, I decided I didn't trust my rotation student.
I should point out that Ted doesn't trust himself either. So
that's true.
That's an important characteristic for good science.
Yep.
So I went about repeating the experiments that he did, which I did fairly quickly because they're relatively easy to do.
Mm-hmm.
And because it works like a maniac.
Yeah, that's true too. So I did the experiments initially in 293 cells because we have those growing at all times, and saw the same twofold-ish phenotype.
So Ted, the twofold is measuring GFP from the lentiviral genome, right?
It's measuring the number of green cells.
Got it.
Post-infection. Yeah, it's not the brightness of the green.
Okay, good.
So
and it's
then
expressed from a lenti vector genome, as you said, yes.
So the amount of green in each cell doesn't go up. It's the number of green positive cells that increases twofold.
Correct.
Okay.
And so when you do that, because it seems to me that if you threw on enough virus, you ought to be able to infect everybody. So you must be using some. It's almost like you're titrating infectious particles. Do you use the same number of particles in the infection and you're not infecting everybody so that you can see the difference?
Yeah, so each one of the experiments that I did was from a titration series.
Okay.
And anything that was above about 30% I would throw out.
Right, okay.
Just because it's, stochastically there's going to be multiple hits in there, so you don't want to...
Right. Okay.
Use that data.
Great.
So you repeated it, and you got the same results, right?
I repeated it and got virtually the same results.
Okay.
Yes.
So what was next then?
And
So
every
then-
time, I should also mention, every time he repeats it, he remakes everything from the beginning. So it's completely new stocks, new transfections, and then new challenges.
Great.
In triplicate, always.
Correct.
So.
Love it.
So then the big question was, could we actually test this in a relevant cell type? And we work with dendritic cells in the lab. Well, not me, usually. But one of the students in the lab, Sean McCauley, every week harvests CD14 cells, differentiates them into dendritic cells, and then does experiments with them. So right around beginning of May time, I decided to grab some dendritic cells from him and challenge those and see if there was a phenotype there. And in the first experiments, I was surprised to actually see that there was a phenotype. It was relatively subtle, but it was certainly there. And that got me excited. But it was sort of a bad time for us because May time for retrovirologists, basic retrovirologists, means we're going off to Cold Spring Harbor retrovirology meeting. So I sort of had this data, went off to the meeting, and then sort of thought about writing this up after the meeting. So did some exploratory stuff with sequences, and I think I grabbed from the database like 1,000 sequences and realized what I was sitting on. These are largely the ones that are in the phylogenetic tree, but I just, using my rudimentary bioinformatics skills, just pulled them out independent of the tree that was put together by Christian. And then we moved on to the whole contacting Pardis and Aaron and having that phone call that, well, I guess Skype call that Jeremy alluded to earlier. And then it was sort of off to the races. So we tested a bunch of different cell lines. We got the bioinformaticians going on things. Sorry, I'm calling it bioinformaticians, but I don't know what.
Computational biology.
Computational scientists and biologists. So yeah, we went full steam ahead from like the beginning of June until the paper came out.
So by this point, you know where A82 is in the glycoprotein, right?
We do, yeah.
Well, tell us just a little bit about where it is and what you think it's doing.
So 82 lies on the surface that interacts with NPC1, the main cellular receptor for Ebola viral entry. And Ebola virus entry is sort of a complicated process. Binding does not occur to NPC1. Binding occurs to, well, depends on the cell type, a bunch of different molecules that are expressed on the surface. And then the viral particles are internalized, and in the endosome, the glycoprotein is cleaved up by cathepsins, and then it's the cleaved form of the glycoprotein that's able to interact with NPC1 and cause fusion. So it's right on the surface of this interface with NPC1, and a structure had just come out around this time. I think it came out in January, that had solved the complex of NPC1 and the cleaved form of the glycoprotein. So I could play around with it and look where 82 is, and it's sort of perplexingly to us at the time, not actually pointed towards the receptor. It's pointed in towards the rest of the glycoprotein. But it was on the interface, so it seemed to be screaming that this was an adaptive change that would allow for interaction to better occur with the receptor.
Mm-hmm. Right. So then you also looked at some other cell types from other species. Can you tell us about that?
And I might just go back a second. As far as the phylogeny and the epidemiology, as best I understand it, and maybe Aaron can comment, the mutants, the viruses that traveled from Guinea to the other countries, which the major countries were Sierra Leone and Liberia, where the vast majority of the cases were, but there also were Ebola made its way to Nigeria and Senegal and Mali, and also to an emergency room in Texas where it was transmitted from a patient to nurses. So all of those viruses that left Guinea, as best I understand it, and Aaron can correct me, have this valine mutation in it.
I think that's mostly true. I know of at least one transmission chain that was towards the back end of the outbreak. I believe it might have been early 2015, where the A82 variant was actually spread from Guinea into Sierra Leone and seemed to infect a handful of cases there. But Jeremy's right on the most part. I think, just because of the number of patients who had the A82 variant versus the 82V variant to begin with, there's just such a huge imbalance. If more people have the 82V variant, then that's the one that's going to be more likely to spread.
Okay.
And then we talked a little bit about the terminology. What do you call the people who analyze these things? And one of the most interesting things about putting this story together for me was interacting with them, because one of the things I realized is that they are computational biologists. I think more than anything, they're actually, I guess you'd call them epistemological philosophers, in that they constantly question what conclusions you can draw from the data you have. And there was just an amazing back and forth among all the authors. What can you say about this? So if all you do is look at the epidemiology of this valine mutation, you could come away thinking, wow, this is amazing. This mutant has completely supplanted and replaced the original virus. It must have some incredible advantage. But if you speak to the strict philosophers, they would tell you, well, it may simply be a founder effect, and it could be a coincidence. And it was a beautiful, constant struggle among us. It was really one of my favorite aspects of doing this project.
Yeah, I think that's great it's unfortunate that part doesn't make it often to the headline writers, Maybe it's a subtlety that's lost on them because, if, as you know, a lot of the headlines say, Ebola became better at infecting people during the outbreak. That was the headline.
Right.
And I would disagree with that.
Yeah.
Although I've not done any computational biology myself, my sense from watching this go on is that this mentality amongst the good ones is very common. Like in analyzing sequence data, it goes through so many manipulations to look for trends and that kind of stuff, and the people who are really on top of it are constantly asking, what does this mean? And in mining the data, that's absolutely the critical question.
Well, of course, in the end, you have to do an experiment, right? Because you can look at the sequence all you want. You have to do experiments. And even then, you may not get a conclusive answer. Anyway, let's get back to TED now. Other cells.
Yep, so we took it from 293s and human dendritic cells. And then we moved into other cells of other species, just what we had laying around in the lab. So I picked a handful of non-human primates, including chimpanzees and rhesus, African green monkey, and then...
Well, wait a minute. I think you might want to preface that by explaining why you chose the species that you chose.
I thought they were just lying around.
Well, that's one way of telling this story. But and also, I should point out that TED tested a good 6 other cell types within human. One cell line in particular, U2OS cells, consistently gave him a 4-fold phenotype, and why that was 4-fold and 293 cells were 2-fold, we don't know. Whether it has to do with expression levels of the receptor or what, but that those were quite reproducible differences. As well, he looked at macrophages. He looked at a number of other cell lines. So whatever it was, he always saw, whatever the magnitude was, he always saw increased infectivity with the valine. Also, in terms of the location of the mutation, which gets to the question of what species he looked at, the mutation, another interesting coincidence, which again could just be a coincidence, that of the 19,000 nucleotides, this mutation happens to be at the point of contact with the entry receptor. There is an alpha helix that is really abutted against the MPC1 receptor, the helix being in the glycoprotein. And the mutation in question is kind of on the back side of the helix, but it's literally there where the contact is made. So while it's pointing in, it's not like it's deep in the protein. It's literally up against the receptor. And structure provided a lot of useful information for guiding the experiments that have gone on since then. And as well, Ted is very versed in looking at phylogenies, and he looked at the structure, or the primary structure, the sequence of NPC1s from different species. And there also was in the background this idea that Ebola exists in some animal reservoir, though there are those like Jens who would contend it may not even be an animal, it may be an insect. Most people,
animals.
Are they?
Yes.
I think Ted said that on the last TWiV also.
He's always keeping me honest. So, but most people think that it's in bats. And anyway, I'll let you take over.
Yeah, I did make it sound like we just grabbed a handful of cells, but there was some thought to it.
Which would have been fine.
Yeah.
Yes. Yeah.
That's a perfectly legitimate reason to use them, but you did have some logic behind it.
we did have some logic, and as Jeremy has alluded to, I'm pretty thorough, and I've done many experiments with a variety of different animal species throughout my time in the lab. So I tend to be a hoarder of these things, and I brought several of them to Jeremy's lab. Jeremy also has some. So this was just a representative panel of cell lines that I had available. But I picked them specifically because we wanted to know if NPC1 was involved. We would expect to see that the sequences that are more similar to human might be similarly more infectious, or the AAD2V would be more infectious in those cells, whereas the more divergent ones, in this case the carnivores and the rodents, wouldn't be. So that's why I picked those that I did. And as Jeremy said, the belief is that the bats, the actual fruit-eating bats in Africa, may be a reservoir species. So the phylogeny that we show is just of the interacting region, or one of the interacting regions of NPC1. And we picked the carnivores and rodents because they have amino acid differences from human NPC1 in the same residues. So we thought that they'd be a good model for bats or other host species, potential host species in Africa.
I wonder if this would be a point to talk about the other paper where the first author is Urbanowitz and the last author is Jonathan Ball, because they did look at bat cells, and maybe you could talk a little about that.
Correct. They did look at bat cells. I believe they looked at 2 species of bats. So if we're going to talk about their paper, I think we should take a step back. They found that AAD2V had a major effect, but they also found other amino acids in the glycoprotein in the context not of AAD2V, but of just the background C15 Makona sequence, could give you a similar enhanced phenotype in human cells. So this doesn't seem to be peculiar just to the AAD2V variant that arose, but there seems to be other ways that the virus could evolve to better infect human cells.
The other changes looked at in the other paper, were they as impressive in the way they predominated from early to late in the outbreak as AAD2V?
No, they were only found in Guinea.
Okay.
Well, the ones that weren't in the AAD2V background were only found in Guinea.
Got it. Okay. So essentially, go ahead, Kat.
Well, I was just going to say, to me, the major point that I was trying to make is that there was this negative correlation between the bat cells as opposed to the positive correlation
with
the primate and cells. Okay.
Yeah, I was going to get there.
Okay.
But they found that there was a negative correlation, and that was independent of background, but dependent upon how well the virus was "quote unquote" adapted to infecting human cells. So, if there was a higher human infectivity for this particular Makona background, then it would have a lower infectivity, a proportionally lower infectivity in the bat cells.
Mm-hmm.
So I thought
which
that
suggests there's a constraint. You can pick one or the other, but you can't necessarily infect both equally well.
Correct.
Right.
And we didn't see that in our hands. So the cells that we tested, they were all pretty much the same between the AAD2V and the AAD2. So there didn't seem to be any phenotype there to be had. But we certainly didn't see a twofold decrease. But again, it's slightly different background for NPC1 receptor.
Now, Jeremy, I remember you saying that, and maybe I'm wrong, but you can correct me. This A82V has never been seen before in any other outbreak. Is that correct?
Well, that's really a question for Aaron, but that's what I've been told.
Okay,
yeah.
Aaron, what do you?
Yeah, that's correct. We haven't seen A82V before in any previous outbreak.
Now, that's sort of why I asked earlier. At what point in this outbreak did A82V arise? And you said after a few hundred infections, and certainly there have been a few hundred infections previously. So that begs the question: why haven't we seen it before? Which probably we can't answer, but I'm sure you have some thoughts about it.
Yeah, I think that one of the thoughts, is just to some degree it's random chance where the mutations hit.
Yeah.
And if you give the virus enough time, anthropomorphizing a little bit, enough time to go through a few human-to-human transmission chains, then at some point, you could get a mutation like this one just happened to kind of win the lottery, so to speak. And especially if that then can travel to a new area with lots of susceptible people, and there's issues with, say, like healthcare infrastructure around there, then it can really spread and really grow quite quickly. Mm-hmm. And so I think that's one of the dangers we want to make sure doesn't happen again for this or other outbreaks. Like, you don't want to be in this scenario where you kind of think, it's just going to burn out by itself like the other outbreaks. We don't have to worry about anything. Let's hide some cases, because it looks bad in the press, and then all of a sudden, just by chance, sometimes things get, we never know for sure that this is what made it worse, but I guess you don't want to take that chance is sort of the point here.
Now, as far as we know, these Ebola viruses with A82V are gone, right? They're no longer circulating in people, and to the best of our knowledge, they're gone. Is that correct?
I don't think that's true.
Why not?
Do you want to take this one, or should I?
Go ahead, Aaron.
Yeah, I would say to the best of our knowledge, we know that they're gone, but I think that, having a lot more survivors now, as well as seeing the appearance of various forms of sequelae, we're starting to understand more that there's a lot more unknown, I think, than known at this point. To point to, there's been quite a few papers about sort of persistence in various immune-privileged compartments. Mm-hmm. One of the most predominant ones being semen, the male genital tract. And I think there's still a lot of questions surrounding exactly what's happening here, but there's been a couple really striking papers. There was one that came out in Clinical Infectious Diseases not too long ago by the European Mobile Lab, showing that it looks, at least genetically, like there was a case of Ebola virus disease. There was a survivor in Guinea who they think possibly through unprotected sex might have transmitted the virus even past 500 days past recovery, which is pretty striking.
Yep.
At least in my eyes, if you'd asked me before I'd seen this, if it's possible, I'd have said there's no chance that just seems incredibly unlikely. But we're seeing all these things now because there's so many more survivors that we wouldn't have seen before. But it's definitely something.
We don't have any sequence from that particular case, right?
Oh no, there's sequence, and so essentially one of the things that's really striking that they show in this paper is that they have a sequence from the survivor's acute sample, as well as from the semen 500 plus days later. And there's actually a few mutations that separate them. But
what about A82? Does it have A82V?
That patient did have A82V, yes.
Okay.
That's correct.
So the point is that there may be other people out there with persistent infections of the eye or the testes who are harboring A82V, right? Okay.
Yeah, there's still some ongoing investigations through a lot of big consortium studies that are trying to assess how common is this sort of phenomenon.
Yeah.
The results haven't really been published yet, but it doesn't seem to be very frequent, which is obviously good news. But I guess this one case highlights that it's possible, and it's something we need to really try and assess how frequent this happens and sort of what's the range of possibilities.
Yeah. So let's assume at some point.
Before we go on, can I jump in here?
Yeah,
Vincent?
of course.
I want to go back to the original question, why haven't we seen A82V before? I don't necessarily think that A82V is particularly special in this regard.
Right.
the other group shows that there's other backgrounds that can give a similar phenotype. We've extended our data a little bit, and we have pretty good data that suggests other amino acid changes could do something similar. So I don't think that we should focus just on this one residue.
Mm-hmm.
Right. That's a good point.
Now, so the other paper where there are other changes that led to increased infectivity, have we seen any of those before? Do you know? You or Aaron?
There's quite a lot of them.
There's many of them, yeah. I have a whole spreadsheet, but I don't have it open at the moment.
So basically there are many ways to potentially increase infectivity, and let's not don't focus on just one. Okay, fair enough.
Correct. And something surprising to me...
Go ahead.
Sorry. Something surprising to me was that there was an implication that some of the amino acid changes were outside of what you would normally consider important regions in the glycoprotein. So they tested everything, including the mucin domain, which we totally ignored. And they found that there were some in like the glycan cap that were important, some that were in the alpha helical region that could be important, and then in the mucin domain that were important. So I think their paper, because of the breadth that they did, suggests that we can't just cherry pick like we did and look at the amino acids that we predict would be by bioinformatics predict that they would be important. We actually need to do the experiments.
Now, on the other hand, you did follow up in your paper with some epidemiological data, which I don't think they had done. Is that right?
Correct.
Yep.
Okay. So they're two complementary papers.
Yeah.
Yes.
Totally.
Yeah.
So the observation of mutations in areas other than the obvious places reminds me of the studies with flu virulence. That you pick up mutations in like the stalk that affect stability and stuff like that. So that's an important point.
Correct.
Yep.
So we were left with this observation that A82V and other changes in the glycoprotein appear to improve infectivity in human cells. So the idea is that it came from maybe a bat reservoir or something else. And then during passage in humans, this change or changes in the glycoprotein were selected for that do better. The problem is that's pretty hard to prove in people. The best you can do is keep observing future outbreaks and see if similar phenotypic changes arise, right? But is there anything else you could do with A82V or any of the other changes to provide more evidence for it helping infect, an animal?
Well, I was kind of a nagging voice throughout this project, and
you always are a nagging voice.
Yeah, I'm and I nagged the philosophers as much to try to with the question: isn't there something in this data that can be analyzed that would indicate that there was some sort of clinical consequence of having this mutation? And the last figure in our paper is, I think, as clear an analysis as one can do with the data that was available. And one has to remember this was I don't know, Dante Inferno kind of horror. I mean, I can't imagine working under the conditions that the amazing people who were there working were working under. There were 800 medical personnel who developed disease from Ebola as a result of working in clinics and hospitals and taking care of people. And 500 of them died. So conditions were not exactly ideal for keeping track of metadata about patients, and all these kinds of things. So that's one really big problem with generating or arriving at conclusions about clinical outcome. The other issue is that if you want to compare the alanine to the valine, you're restricted to only a subset of cases from the beginning of the epidemic, because that's the only time really when the alanine was present. Right. And so we were left with somewhere in the order of 200 total cases from Guinea, which is the only place really where the alanine was from the beginning of the epidemic, from controlled as best we could. So there's maybe 120 valine cases and 70 or so of the alanine cases that were compared. What I was really hoping we could find something within the viral load data. And so viral loads were measured. Again, this is a subset of patients on which that data was available. And there seems to be a trend for a higher viremia, though it's not statistically significant. Higher viremia associated with the valine mutation. What was significant was an increase in the mortality of those who were infected with the valine. They're not, I guess, as Vincent has pointed out elsewhere, they're not gigantic effects, but I think they are reasonably convincing. Can we say that they were necessarily due to the mutation? We can't. It's an association. There may be all kinds of confounding variables in terms of how long people took to get medical care and all these other things. So it's not the cleanest of data, but it seems like there's a trend there. I don't think from the epidemic there's going to be anything more conclusive than that. That analysis in terms of the clinical significance of this mutation. So you asked what one could do going further. Yep. Again, a philosopher might say there's nothing you can do. I, as a virologist, pretty concrete in my approaches, I would say there are experiments we could do that I would find reasonably compelling, depending what results we got. And indeed, so a caveat to both of these papers. Well, so that the good thing about both of these papers is that they are the first actual hard experiments in controlled settings that have been done with these mutations, and that's why I think they're so important. So until now, there's been a lot of computational analysis. Going forward, though, all of the experiments that were done, they utilized 3 different pseudotype systems. They used an HIV pseudotype, a murine virus pseudotype, and they used an Ebola VP40 core structural protein pseudotype as well. And the results were the same with all of those. The conclusions were the same. But none of these used full infectious Ebola. And so going forward, I think really critical will be to generate the proper reverse genetic strain that corresponds to the ancestral virus that gave rise to these mutations, and then a comparison between the viruses derived from that system, wild type, or with these variants that appeared over the epidemic. And I think meaningful experiments could be done in tissue culture. Just do you see replication effects in tissue culture with these mutants? Right. And then where do you go from there? So the standard in the field, I guess, are non-human primates. My my personal preference would be to do experiments with so-called humanized mice, where much of the pathology has been reproduced in the small literature that exists so far using full Ebola challenges there. So these are mouse strains that are genetically defective in terms of their immune response, and they are reconstituted with a human xenograft of immune hematopoietic stem cells, and these can generate a new a human immune system within the mouse. And these animals are infectable only when they're reconstituted with these human cells. And it is a lethal infection only when they're reconstituted with the human cells. So in that context, one could have a small animal model where one could actually look at transmission. The human cells, in fact, reconstitute the mucosa. You could even do mucosal challenges. And you could look at such issues as what's the viral load when I challenge with these different viruses. If I do a competition between these viruses, which virus wins, which limiting dilution, which is more infectious, which is more pathogenic, and one can sequence over the course of the infection and see what evolution occurs in whatever virus you're challenging.
I totally agree, and I think the real key here is, do these these effects, these twofold effects that you see in infectivity and cell culture, would they have any impact in pathogenesis, transmission, viral loads, et cetera? I think it's important to know that. You've mentioned that in your paper, and I think that's key. Get out of the pseudotype and into real bona fide virus and animals. Now, that's not something you can do there because you need a BSL-4 lab. So
right.
Are you trying to convince someone to do it with you?
Yeah, we've been in discussions for. As soon as we found out about our phenotype, I started speaking with the people who are in a position to do these experiments. And, as I understand it, Portis was having the same conversations way before we got involved. And there's, I think on a certain level, you can understand the resistance. They're big experiments. They're expensive experiments. You have to convince someone that a single alanine to valine mutation in the glycoprotein is going to be significant. A lot of the algorithms that people use to predict functional changes in proteins or significance in terms of the evolution of the virus didn't really score very highly. But
now you have functional data, so that should be convincing, right?
Yep.
Yeah, so immediately after our paper came out, we heard rumors that there might be movement. So I'm hopeful that something will happen.
Well, I'm visiting Rocky Mountain Lab next week, and I'm doing a TWiV with Heinz Feldman, and so I'll ask him if they're going to do this. Heinz Feldman is the Ebola guy out there, so I think that's important. Now, Jonathan Ball is the senior author on the other paper that we've mentioned, which is called Human Adaptation of Ebola Virus During the West African Outbreak. And I have to say, I like your title, Jeremy, Aaron, and Ted, much better, which is Ebola virus glycoprotein with increased infectivity dominated during the epidemic. This human adaptation is an assumption. And I think that's where a lot of the offending headlines came from, that title. Anyway, I wrote a blog post on this, and Jonathan emailed me, and in my post I said, would be nice to know in an animal model what this change does. And he said, quote, "How would animal models tell us about the impact of these mutations on humans? I would be cautious in interpreting the relevance of macaque or glycoprotein models as perfect surrogates for human infection. Of course, it doesn't predict anything, but you would like to know whether these changes have any effect on pathogenesis in any animal model, right?" That's what you've been saying,
yes.
Anyway. So I think Jonathan, in the end, wouldn't agree with me he said, "We will never agree on this." Just unfortunate. So I said, "Why don't you come on TWiV and talk about it?" And at which point he stopped communicating with me. And as you see, he's not here today. So I think it's very worthwhile to do these experiments simply to know if these changes have any effect. Because if you find that they have no effect on viremia or transmission or anything, then you wonder what was their selection in the first place. So,
right. Well, so let's see.
Is Jonathan a philosopher or a wet?
No, he's like me. So
it's like you. Okay.
Yeah. So we commiserated ultimately when we realized we had similar stories, and we decided to submit our manuscripts together. He and I commiserated about the suffering we endured at the hands of these philosophers. In fact, Jonathan pointed out, so we talked a lot about this word adaptation, and I have to, in defense of Jonathan, he was a wonderful person to communicate with, and I think his paper is amazing. And I have to say that he and I both like the word adaptation. We both, as I think I mentioned to Carl in the article he wrote in the Times, in my view, if it looks like a duck, why not call it a duck? So while we can't prove it is, it wasn't just a founder effect. Strictly speaking, we can't prove that. I don't know what we can prove in biology. It does indeed look like an adaptation. And if, as Jonathan pointed out, he actually scanned our manuscript, and he mentioned that we used the word adaptation 16 times in our manuscript. But every time I wrote it, one of the philosophers would come in and qualify it. So it always has some modifier.
Yeah. So I'm looking at one here. If GP82V is an adaptation, then, it's very, very qualified. And then you end up saying, we can't make any conclusions about this. I think that was appropriately, but that gets lost with Carl Zimmer. He doesn't write that part because he doesn't want to. He wants to write it adapted to humans, right? Well,
I don't know that I agree with that, but I think it's a very fair presentation.
Well, I think that that it's about shocking people and telling them, "Wow, this is what happened." But because if you say, "Well, we don't know this and that," then it's wishy-washy, and as Alan would say, it's not news then, right?
Well, yeah. I mean, there's kind of a fine line. Oh, the newsworthy part of this is that it's, why is Cell publishing it, right? Because this is Ebola, and you've done some interesting science on it, and you found something that is kind of looks like an adaptation, and you want to come out and say that. And there's only so much. There are only so many of the caveats that you can put in the piece that you write for the general audience. Carl generally does a pretty good job of that, but where do you draw the line? And as Jonathan Ball's letter indicates, this isn't even. There isn't even agreement among scientists over where we draw the line and whether this is an adaptation.
Yeah. Well, I think NPR did a good job. They were, let's see, the headline, "Mutant Ebola may have caused outbreak." That's pretty good.
Right.
Anyway, I think that what Carl wrote was an amazing piece of text. I'm totally amazed by it, considering how difficult I found it writing the story and combing through the various press releases from our universities, rewriting them, and trying to convey things clearly, and not trivializing things. I don't know. I thought it was very good, and I also thought. He did confer with someone who published a year before that there really was no evidence for any kind of significant adaptation on the part of the virus. He actually quotes them in his article. So I think he did a masterful and very professional job, actually.
And let's also put this in context. When this outbreak was ongoing, we had folks writing editorials about how the virus was going to mutate because it was infecting so many people, and it was going to become airborne, and all sorts of horrible things were going to happen. And of course, none of that came to pass. And what we end up finding out is that there are these mutations and these lineages that seem to have become predominant during the epidemic, and there's some evidence that some of them may have been adaptive, and that's the kind of thing you'd actually expect, not the blown out of proportion, now it's going to spread like the flu type of nonsense.
Right. Well, I think similar things are happening with Zika virus, right? There are lots of sequence analyses being published and claims by the philosophers that these are accounting for the phenotypes, but now we need to have some experiments done like these.
Right.
It's important to do.
That's true.
I mean, this is an interesting problem because, in terms of adaptation of viruses, so in zoonotic spread, the virus goes from an animal, non-human animal to a human. The assumption is there's some kind of adaptation that goes on, selection of mutants that make the virus in the end a human virus, like SIV becoming HIV. And the problem is we never could get a handle on this because SIV was so long ago, that transmission. Measles was thousands of years ago. MERS, we don't have a clue. Even Zika, we don't have the original viruses. So this is an example where you have these series of isolates throughout the outbreak. It's the first time I can think of that we might have a handle on at least some ideas of what changes could in principle lead to better adaptation. But again, it's not proven in the end by the standards of experimental virologists anyway. But I think it's suggestive. So Jeremy,
the other point in Jonathan's defense regarding adaptation is his argument that if anything, there's a cost to the virus on the putative reservoir host, which I think is really the piece of evidence that emboldened him to use the word adaptation.
I would say it's adaptation to human cells, right? You have not done the experiment in humans, and that's what the title says, and that's not right. Adaptation to humans is not right. It's selected during, and you said it could be a founder effect. It's selected during passage. It has a phenotype in cells, but I don't think you can say adaptation to humans, not yet. And I don't know if you ever could, actually. So, someone could argue, well, if you can never prove it, why not just say it? And I think you were basically saying that. But I'd like to be rigorous about it.
Well, to be in his defense, it doesn't say adaptation to humans. It just says human adaptation. And so
that's the same thing, isn't it?
Well,
I'm not
maybe not. But okay. And then in Carl Zimmer's sub-headline, which maybe he didn't write, its mutated virus was more effective at attacking human cells.
Human cells is okay, but not every headline did that, as you know. Many of them were far, far worse.
So the other point that I think we should discuss here is that we do not know where the reservoir is.
Right.
And it seems to me right now that's the single most important question. I mean, where is it hiding? Why are these sporadic outbreaks occurring? Jens argues it sure as hell ain't in bats. It's something else.
Mm-hmm.
He has a whole. He said he hasn't been on your show on TWiV for a year, and he has to come back and do his rant. But,
okay. No problem.
But until we really have a handle on that, we can't address this adaptation question because we don't even know what it's adapting from.
I totally agree. Totally agree. Yeah. And then Jeremy, you plan to go to Africa and collect samples, right?
At some point, yeah. Yeah.
Is there anything else? I want to finish in the next 20 minutes or so. Is there anything else that?
I actually.
Go ahead.
I've been wondering whether you could do a biochemical assay for the binding to back up the pseudotyping experiments.
Good question. I can answer. Ted, do you want to take a stab, or do you want me to do it?
I could take a stab, but I don't know. I think the best way to answer it is yes, hypothetically you could. And I think some of the popular press comments highlighted things Jeremy said, that there's this possibility that it's just strictly binding to the receptor that's changed with the A82V, and then there's also the possibility that the mutation has made it easier for the glycoprotein to undergo the conformational changes that allow for exposure of the fusion peptide and all that.
Okay, so even if you could do a binding experiment and didn't see any difference, that wouldn't necessarily give you tremendous insight into what was going on.
Exactly.
Right.
Because it could be something else. Okay.
Yeah, and so in fact, there's a lot more underlying Ted's comment. He's done a lot more work. In fact, he's generated about 50 mutants, just engineered them based on the crystal structure and a model of what we think is happening. So our idea is that in fact, it's not increasing direct affinity for NPC1, but that it's making the conformational change much more likely. And there are all kinds of detailed arguments based on the structure of the molecule we use to make that argument. And Ted has a number of mutants now that support his model. We've also heard rumor that someone else out there may have actually done binding experiments and not seen anything. So we even started to set up to do the binding experiments, and we decided that it probably was not, it didn't fit with our understanding of it, and so we didn't pursue that, which we thought that they would be pretty difficult experiments anyway.
All right. Anything else from anyone before we move on? Aaron or Ted or Jeremy, anything you wanted to say about the study before we leave it? I think we've covered it pretty well, but last chance.
I just want to pipe up and say how great this study was to actually perform. I've participated in many collaborations over the years in the labs, and this has been one of the most rewarding. It was at every turn great to interact with all of the people on our paper as well as the people from Jonathan Ball's group. I was pleasantly surprised at how everything went during the whole process.
All right. Aaron, anything else?
Yeah, just ditto on my end. It was so easy working with these guys and bringing in so many different expertises. I thought was something that's really cool.
I presume you're continuing to work on this as well, Aaron?
Yeah. So we've got some other studies in the work looking at some other mutations as well. So
in other parts of the virus.
Other parts of the virus? Okay.
Yeah. So one of the interesting things is there were very few non-synonymous mutations that really stuck, that established clades, and that has kind of made it easy to figure out what to work on from the virology standpoint. So there are a couple others out there that Aaron's working on, and we're running along with him on those. I have to say, I agree completely with what Ted said. It was an incredible experience just working with this group of people. There really was nothing. There was no way that any individual could have done this project. We had such different perspectives and such different expertise, and we relied so much on each other. It was really enjoyable, must say.
All right, cool. Aaron, I just wanted to ask you: is your lab interested in Zika at all?
Yes, we've actually been doing a little bit of work in terms of the sequencing. It's been fairly challenging, and we've started to overcome some barriers in terms of just getting sequencing data from clinical samples. But we actually just published not too long ago a fairly large release of Zika virus genomes from the outbreak from a number of sites across the U.S., Central, and South America. And for anybody who's interested, these all get uploaded to a site, www.virological.org. And there's a lot of other viral genomicists kind of out there on the site. So there's data from our group as well as many others too.
All right. For people who want to do phenotypic studies.
Yep.
There
That
you
would
go.
be. Yep.
All right. Let me tell you about the sponsor of this episode, CuriosityStream. It's the world's first ad-free nonfiction streaming service. They have over 1,500 titles, 600 hours of content, founded by John Hendricks, who used to be with Discovery Channel. So you're going to get access to real science shows. It's available on the web or on any of the devices that interface the web with your TV, like Apple TV, Roku, etc. 196 countries worldwide, and they have a wide variety of science, technology, nature, history, interviews, lectures. If you go over to their site, curiositystream.com, just search for science, you get a ton of really interesting stuff, like, here's a cool one, Stephen Hawking's Favorite Places. It's a brand new documentary in which he pilots a CGI spaceship across the universe, making stops at many of his favorite destinations. And there's Digits, a 3-part series hosted by Derek Mueller, who created the YouTube science channel Veritasium, has interviews with Edward Snowden and Vint Cerf, Deep Time History, story of the universe's 14 billion year history, much, much more. A lot of great stuff. They also have a lot of super high definition 4K content. They have over 50 hours of that. They have monthly and annual plans available, starting at $2.99 a month, less than a cup of coffee. Check out curiositystream.com/microbe. Use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 entire months free of one of the largest nonfiction 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign-up. We thank CuriosityStream for their support of TWiV. Let us go directly to picks of the week. I have a hard out at 1. We're doing a TWIP, so we don't have time for letters today, but we'll do them next time. Let's see. Who should we start with? Let's start with Jeremy. What is your pick of the week?
So I picked real-time tracking of Ebola virus evolution, which you can, there's a link I guess we can put in for the website. It's an amazing way to visualize the evolution of the Ebola virus over the course of the epidemic. They have a similar kind of viewing tool for the Zika virus as well. You can click on different parts of the genome and look within a phylogenetic tree and see where that mutant is in the course of the epidemic. This was built by 2 people, Richard Neer from Max Planck in Tübingen, and Trevor Bedford, who's at the Hutch in Seattle. And it's really amazing. It's worth taking a look.
Nice.
Cool.
Both Richard and Trevor are on Twitter. We will put their links in so you can pop questions. They're philosophers, right?
They are that kind of scientist, yes.
Is that a new word?
We have a new arc.
That's
we have
right.
a new word.
Philosophers.
Yeah.
Right.
Aaron, what do you have?
I have a pick that's space-related, going back to a common theme. This was a little bit before my time, but one of Pardis's close friends, who is a microbiologist turned astronaut, Kate Rubins, went up into space to the ISS to do sequencing of DNA in space, just to see if it would work or not, with the hope being, if they find any kind of organisms or genetic material in space on some planet, they might be able to just sequence it there directly. So there's a couple links from some
so
of the
they know
NASA
whether to
webpage.
nuke it from orbit, right?
Yeah. Plus a really cool video interview that she did actually with the Broad community, as well as the folks, the Whitehead, which is just next door to us, and MIT, which are kind of all in the same area. It's really cool. It's pretty wild. She talks about a lot of the things that are similar and maybe a little different about doing bench work in space. Sounds like she's got a really cool setup, and I believe she landed safely a week ago or so.
Cool.
But yeah, it's really cool. You should check out the video.
This gives new meaning to the phrase sequence space.
Yeah.
Yes.
Aaron, what was the website with the sequences on that you had just mentioned? the Zika sequence?
It's virological.org.
Virological?
Yeah, I can add a link in the show notes.
Okay, great. Thank you. Ted, what do you have for us?
So my pick is YouTube channel called The Brain Scoop. It's hosted by Emily Grassley. And I think this might have been picked based on the notes by Kathy previously. But it's a really good channel. She does a good job of conveying the importance of museum collections, how they're made, what they consist of, what you can infer from them, and why it's important to preserve them for future researchers. And I think because I'm sort of warped, I really like it because she has a wicked sense of humor.
Yes.
So I find it very entertaining, even though it's peripheral to the discussion at hand.
Great. Yeah, it was picked, but it can't hurt to pick it again.
That's right.
Yeah, I picked it back when the specific episode I picked was about women in science. And so that's the link that I provided. It was in 263.
So on our recent TWiV, we had a guest from the Field Museum, and she knows Emily because Emily works at the Field Museum. They hired her to make these videos, which is really cool. Nice job. All right, thanks, Ted. Alan, what do you have for us?
I have something. Speaking of things that have been picked before, Dixon actually picked this. I searched back through. He picked it on TWiV 252, I think. And I had come across that mention of it, and it had been mentioned a couple of other times in other things that I'd read. And every time I thought, oh, yeah, I ought to get around to reading that. It's a book called A Sand County Almanac by Aldo Leopold. And this was published in 1948, I think. Aldo Leopold was the founder, really pretty much the founder of modern wildlife management biology. And the Sand County Almanac section of the book is just a month-by-month telling of what's going on this abandoned farm that he has bought and that he's allowing to return back to nature in Wisconsin. And he discusses the interrelationships of all these organisms and what this used to be like and what it's like now and why. And then in the next part of the book, it's a collection of additional essays where he essentially lays out the concept of a land ethic, which when you read it now, a lot of it is, well, yeah, of course, But the reason you say, yeah, of course, is because of this book. He was the one who developed a lot of these concepts, building on earlier things, but really bringing it all together into a biological view of biological and ethical view of how we ought to relate to, or how we ought to think about relating to the land and the environment. And it's a lovely read. His writing is gorgeous, and I'd highly recommend this to anybody.
It's classic.
Excellent.
Great book. Yep. People need to read it. Rich, what do you have?
I also have a repick. This comes under the heading of technology and just general nerdiness because I'm a sailing enthusiast. But I'm picking the Vendée Globe.
Yes.
I picked this 4 years ago. I've talked about this on more than one occasion before. This is a sailboat race that is around the world, nonstop, unassisted, single-handed. These guys are nuts.
Yes.
They took off 3 days ago, 29 skippers in these boats that are specially made for this race. They're 60 feet long. They're like giant surfboards designed to surf down 40-foot waves in the Southern Ocean. Okay? And it's crazy. There's a website that you can use to follow the race and keep track of it on a map. And the skippers post videos and comments. So if you get into it, you wind up following a skipper or two or three and watching the whole thing. It's going to take them 3 months. And you'll hear from me now and then to, update you on what's going on. But,
and what's so nutty about this is not only are they driving these massive boats at ridiculous speeds through these insane weather patterns, but the boats are built absolutely as lightly as they possibly can be without falling apart. You hope.
Which they do occasionally.
Which they do regularly. This stuff is constantly breaking on them, and then they're really in a place where they can't be rescued. It is a crazy, crazy race. This is fun.
So get hooked. Follow it.
All right.
Unfortunately, the website's a little flaky. It's not the greatest, but so you have to be patient with it, but it's worth it.
Yeah.
All right, Kathy, what do you have?
I picked a link to a blog post that talks about something that's in the ecology and evolutionary biology world called Axios Review. And one of the blog writers is on the board of this review. And essentially, authors can send their papers in to get pre-reviewed by expert reviewers that are chosen by an independent editor. And the authors can also then specify journals that they're thinking about submitting it to. And so then they can get the reviews back, and the editor suggests what the journal might be, and if they want, they can even then forward the paper to the journal with the reviews and ask the journal if they want to have would they be interested in seeing the revised manuscript and accepting the paper. And it's just a totally foreign concept to me when I first read about it, and I've mentioned it to a couple people, and they kind of react on all ends of the spectrum. Like, no, I would never have somebody tell me what journal to send my paper to. But it's just an interesting concept. It's a nonprofit thing now, and there's an example of the blog writer himself having done this, and then some interesting comments.
They're like scientific literary agents.
Mm-hmm.
Very interesting.
Yeah.
Very interesting. All right. My pick is a website of a photographer, Lowe's Heerink. And in particular, one part of his site, he does photographs from above. He calls them vendors from above. He's in Hanoi, and he's taking pictures of people carrying various things on their bicycles. They are just beautiful.
These are neat.
I love them. They're all very similar because that's the shtick, right? But they're all different flowers, vegetables, just gorgeous. So check it out. He has other stuff on his site also that's pretty neat. Vendors from above.
Are you sure? Are you sure that this is
I'm not sure of anything.
Because there's self-portraits here.
Well, I didn't look at those.
Huh.
Why aren't you looking at the bloody?
Because I looked at the whole thing because they're wonderful.
Me too.
I looked at all this stuff.
It's a woman,
and this is a woman.
All right,
yes.
It's a woman.
Although there's a heading below that called gender dysphoria, but
yes,
it's not popping up with any photos, so I'm not sure what that means.
So she takes overhead photos of vendors, which is my pick. Vendors from above, they're really cool. No listener picks today. That'll do it for TWiV 415. You can find it at iTunes and microbe.tv/TWiV. Send us your questions and comments. We like getting them. TWiV at microbe.tv. And consider supporting our work. Go to microbe.tv/contribute. We have a variety of ways to help us out financially, including Patreon. Our guests today have been from the University of Massachusetts in Worcester, Jeremy Luban. Thank you, Jeremy.
Thank you, Vincent. It was fun.
Jeremy's at Luban Lab. That's your Twitter, right?
Yeah.
Luban Lab. Ted Diehl is also at UMass Worcester. Thank you, Ted.
Thanks for having me again.
Our pleasure. You do good work. You get on TWiV, you know. That's the way it works. Ted is at...
Try to do that again.
Yeah, I'm sure you could. Ted is at Viruses Are Us. Viruses_R_us. Aaron Lynn is at the Broad Institute of Harvard and MIT. Thank you, Aaron.
Thank you for having me. It was a lot of fun.
Hope you enjoyed it, and we'll come back one day when you have more cool results. Told your boss that we'd like to get her on sometime. She seemed
definitely.
She seemed to think it would be cool. So
100%.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Residing in Austin, Texas. That's okay.
Whatever.
You're welcome. And Lowe's is a girl's name. Okay. Always a good time.
Thank you very much. Lowe's is a girl's name. Alan Dove is at alandove.com. You can also find him on Twitter. Thank you, Alan.
Thanks. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsor of this show, CuriosityStream. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.